Technical Veterinary Learning. Founder and producer is Professor Ahmed Mamdouh El-Sherif, Professor of Epidemiology and Infectious Diseases, the ex-head of Department of Veterinary Medicine, the ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University. We will speak today about babesiosis, Texas fever, red water fever, cattle tick fever. All of them are synonym to the name babesiosis disease. Babesiosis is an infectious disease of ruminants, equines, wild animals, and pigs of tropical and subtropical countries all over the world. Babesiosis characterized by anemia, hemoglobinuria, jaundice, fever, and high cases fatality rates. Diagnosis of babesiosis. Epidemiology of babesiosis all over the world. Number 1. Pathogenic risk factor. Family Babesiidae, genus Babesia, order Pyroplasmida, phylum Apicocomplexa, with different Babesia species that infect different animal species. The nomenclature of Babesia as intraerythrotic parasite is still subject to change. Different Babesia species will infect different animal species as the following description. For cattle species, there are 3 different Babesia species. Number 1, Babesia bovis or Babesia argentina. Number 2, Babesia bigemina. And number 3, Babesia divergens. This is the shape of Babesia bovis under microscope with staining with Giemsa stain. This is the shape of Babesia bigemina under microscope with staining of blood film with Giemsa stain. This is the shape of Babesia divergens under microscope with staining of the blood films with Giemsa stain. Babesia species that infect sheep species are 2. Number 1, Babesia mutesi, and number 2, Babesia ovis. The shape of Babesia mutesi under microscope for blood films stained by Giemsa stain. The shape of Babesia mutesi under microscope from blood films stained with Giemsa stain. Shape of Babesia ovis under microscope in blood film stained with Giemsa stain. Babesia species infect equine are Babesia cabali and Babesia equi. Babesia cabali under microscope. Babesia equi under microscope. In case of wildlife animals, there are 3 species of Babesia. Number 1, Babesia odocelli, which infect white-tailed deer, American elk, American woodland caribou, desert bighorn, sheep and red deer. Number 2, Babesia divergens of reindeer. Number 3, Babesia trandifringis of reindeer. Babesia odocelli under microscope. Number 2, occurrence of the disease. Distribution of the risk factor Babesia species is governed by geographic and seasonal distribution of the insect vector that transmit them. And this is changes all over the world due to the changes that occur in the atmosphere all over the world. Distribution and occurrence of Babesia bovis countries all over the world: Australia, India, China, all east of Asia, Saudi Arabia, Egypt, Sudan, and the east part and south part of Africa, Algeria, Morocco, and some countries of the West Africa, some countries of the south of Europe, such as Turkey, Spain, Portugal, France, all the countries of Southern America, except parts of Argentina, the middle part of America. Occurrence of Babesia bigemina all over the world countries. It is the same like distribution of Babesia bovis countries. Occurrence of Babesia divergens all over the world countries are Turkey, some parts of Italy, then Spain, Portugal, France, Germany, UK, and Scandinavian countries. Susceptibility to babesiosis. In all animal species, animals under 6 months showing lesser infection than adults due to colostrum immunity. But ages between 6 and 12 months showed a higher infection rate due to the decline of the immunity. Origin of infection and transmission of babesiosis. Variable stages of Babesia species are present in the bloodstream of animals in the active phase of the infection. Ticks are the natural vectors of babesiosis. The right of the screen denoted to the shape of a tick after engorged by blood after a meal. Number 3, different tick species are Rhizophilus, Bophiolus, Rhibophilus, Hyalomma species, and Iodix species, and this the shape of them at the left part of the screen. Life cycle and pathogenesis of Babesia species. Life cycle and pathogenesis of Babesia species passes in 3 phases. Number 1, asexual reproduction in ticks. Number 2, infectious and sexual reproduction in animal species. Number 3, destruction of Babesia organisms that infect the RBCs in the spleen. This is the explanation of life cycle and pathogenesis of babesiosis inside the ticks and animals. The first is asexual reproduction in ticks, where is gametogenesis, 4 zygotes which occur in the gut of the ticks, that it changes into cnids, passes with the hemolymph. Then cnids changes to sporozoites that multiplied in the salivary gland of ticks. Then when the tick take blood meal, will transmit this infection to the bloodstream. When the sporozoites enter the bloodstream of any animal kind, it will be developed a sexual development, as Babesia infect the RBCs. The Babesia organisms will be destructed in the spleen, causing splenomegaly, as appear here with sonar detection. Sources of infection to babesiosis. Sources of infections: number 1, tick bites. Number 2, contaminated surgical instruments and syringe needles. Economic importance of babesiosis. Economic importance of babesiosis: number 1, direct losses of production, either for milk production or meat production. Number 2, restriction of movement of cattle for trade, as diseased cattle will be treated for a week and then convalescent take another a week to 10 days, so restriction of movement will be almost 1 month. Number 3, many infected animals will die during the acute phase of the disease, especially in epizootic areas. Number 4 of economic importance of babesiosis is the long period of convalescence, which reach to 14 days. Number 5, major meat and milk production losses. Number 6, incidental costs of immunization against the disease and the treatment of clinically diseased animals will be added to the economic burden of this babesiosis. Number 2, clinical symptoms of babesiosis. Number 1, cattle infected by Babesia bigemina are showing hemoglobinuria that present early and more constantly. Number 2, fever condition is less of a feature of babesiosis caused by Babesia bigemina in case of cattle. Number 3, recovery in neonatal cases is usually rapid and complete. Clinical symptoms of Salmonella abortus ovis infection. Number 1, abortion during the second half to last third of gestation period. Number 2, lambs may be also still born or dying within the first day of life. Number 3, mortality in lambs is common from either weakness, ensuing hypothermia and or hypoglycemia, or due to development of acute pneumonia in previously healthy lambs up to 2 weeks old. Number 4, in case of clinical symptoms of cattle that infected by Babesia bigemina, maybe the disease can develop very rapidly with sudden and severe anemia, jaundice, and death. Animals that recover from Babesia bigemina infection remain infective to ticks for 4 to 7 weeks. Animals that recover from Babesia bigemina remains as a carriers for only a few months to other cattle. Cattle infected by Babesia bovis species will showing: number 1, acute disease generally runs a course of 3 to 7 days. Feverish condition is greater than 40 centigrade or 104 Fahrenheit, is usually present for several days before other signs emerge and present. Number 3, feverish condition followed by inhabitants, depression, polypenia, weakness, and reluctant to move. Rest of clinical symptoms for cattle infected by Babesia bovis is faces. Number 4, hemoglobinuria is often present, and the disease is known as red water disease in some countries. Number 5, urine is dark red to brown in color and produces a very stable froth. Number 6, anemia and jaundice develop, particularly in prolonged and severe cases. Number 7, diarrhea may occur. Number 8, muscle wasting, tremors, and recumbency develop in advanced cases, followed terminally by coma. Number 9, many severely affected animals die, particularly at this point after an illness of only 24 hours. Number 10, in case of clinical symptoms for cattle infected by Babesia bovis infection, metabolic acidosis can be present in a significant percentage of cases of bovine babesiosis. Number 11, during the fever stage, pregnant cattle can abort and bulls may become sterile for 6 to 8 weeks. Number 11, cerebral babesiosis is manifested by incoordination followed by posterior paralysis or mania, convulsions, and coma. The mortality rate in such cases is high despite chemotherapy, either beginning at the end of the cases or near end of the cases or at the middle of the cases. Number 13, in clinical symptoms of cattle infected by Babesia bovis, those cattle that survive their febrile stage usually lasts for approximately a week, and the total course of the disease is about 3 weeks. Number 14, cattle that survive recover gradually from severe emaciation and anemia, which are inevitable sequelae. Number 15, subacute syndrome also occurs, particularly in young calves, in which fever is mild and hemoglobinuria is absent. Clinical symptoms of cattle infected by Babesia divergens. Number 1, the syndrome associated with Babesia divergens is like that of Babesia bovis. Number 2, there is spasm of the anal sphincter causing the passage of feces with great force in a long, thin stream, even in the absence of diarrhea. This sign is referred to as a pipe stem feces. Clinical symptoms of sheep species infected by babesiosis are fever, anemia, icterus, hemoglobinuria are common signs. Clinical signs of babesiosis in wildlife animals. Number 1, babesiosis reported during periods of stress such as rutting seasons, coughing seasons, transportation, and or overcrowding. Number 2, lethargy symptoms. Number 3, hemoglobinuria. Number 4, icterus. Number 5, fever and recumbency. Number 6, sudden deaths will be reported in Cases of wildlife animals infected by babesiosis. Ecteric vaginal mucous membrane as a clinical signs of babesiosis in naturally infected cow showed here in this video. This figure showing heifer cow with ecteric skin and vulval mucous membrane. This figure showing buffalo calves more than 12 months in age that showed enlarged inguinal lymph nodes and stunted growth in the upper part of the figure. In the lower part, there is blood film stained by Giemsa stain showing the Babesia species organism inside the RPCs of them. This figure showing large number of ticks on cow's dewlap, as these ticks act as a source of transportation of Babesia species for several generations through trans-ovarian theory. This figure showing a case of hemoglobinuria in a cattle suffering from babesiosis. This figure showing a cattle with an enlarged inguinal lymph node due to infection by babesiosis. Emaciated cows due to long-term infection by babesiosis. Laboratory diagnosis. Samples collected for diagnosis of babesiosis are: number 1, blood sample; number 2, serum sample; number 4, biopsical samples from lymph nodes; number 5, ticks hemolymph; number 6, parenchymatous organ of dead animals; number 7, bone marrow. Laboratory tests for any case of babesiosis: number 1, microscopical examination of blood-stained smear; number 2, tissue and hemolymph smear from ticks that present on infected animals; number 3, serology tests such as complement fixation test, ELISA test, and indirect fluorescent technique; number 4, hematology RPCs count, including BCV and hemoglobin percentage; number 5, PCR and phylogenetic tree document; number 6, transmission tests. Microscopical examination. This is the shape of Babesia bovis under microscope with staining with Giemsa stain. This is the shape of Babesia bigemina under microscope with staining of blood film with Giemsa stain. This is the shape of Babesia divergens under microscope with staining of the blood films with Giemsa stain. The shape of Babesia mutaceae under microscope for blood films stained by Giemsa stain. The shape of Babesia mutaceae under microscope from blood films stained with Giemsa stain. Shape of Babesia ovis under microscope in blood film stained with Giemsa stain. Babesia cabali under microscope. Babesia equi under microscope. Babesia holocelli under microscope. PCR test. Real-time PCR detection kit Babesiosis. Item code GPCRS. Babesiosis technology. Real-time PCR. Manufacturer GPCRS. Variant target 100 reaction. Monodose target 24 quantum PCR tubes. Representative gel showing the specificity of ssRNA PCR for the efficient detection of bovine babesiosis. Lane 1 is the negative bovine genomic DNA control. Lane 2 is the positive control for Babesia bovis. Lane 3 positive control of Babesia bigemina. Lane 4 is a positive sample for Babesia bovis at 541 base pair, and lane 5 positive sample for Babesia bigemina at 1124 base pair. Lane M is 1000 base pair DNA ladder marker. Electrophoretic detection of Babesia species PCR amplification products or agarose gel, where it ranged from 100 up to 500 base pair molecular weight marker, and negative control is about 420 positive Babesia species fragment of 18S RNA gene is line 9, while line 1 up to 11 are negative samples. This figure showing blood smear from Babesia infected sheep was stained by Giemsa. Giemsa stained blood smear from large Babesia A and B, and from small Babesia C and D. DNA was isolated from the blood smear of Babesia infected animal and analyzed by PCR. PCR analysis with primers B1, B2 specific for 18S RNA gene of Theileria and Babesia. Lane 1 DNA from small Babesia. Lane 2 DNA from large Babesia. DNA from small Babesia was analyzed by PCR with primers B1, B2, lane 1. The corresponding PCR product was analyzed by semi-nested PCR using Babesia bovis specific primers B3 and B2. At lane 2, DNA from large Babesia was analyzed by PCR with primers B1 and B2 at lane 3. The corresponding PCR product was analyzed by semi-nested PCR using Babesia bovis specific primers B2 and B3 at lane 4. The PCR product with primers B1, B2 was cut with restriction endonuclease, where small bovis Babesia at lane 1 and large bovis Babesia at lane 2 and 3. Lane 4 from uncut PCR product. M is remarkable to 100 base pair marker. This figure showing the phylogenetic tree of Babesia and related pyroplasms with zoonotic species bolded. The tree was constructed using neighboring joining analysis of full-length 18S RNA gene sequences extracted from the gene bank. This figure showing the phylogenetic tree for the complete 18S RNA gene from selected Babesia species. Babesia cabali, Babesia bigemina, Babesia species from Post-mortem, Babesia bovis, Babesia gibsoni Asia1, Babesia diversion, Babesia odorisi, and Babesia species from Europe. The tree was computed by using the quarter closing maximum likelihood method of tree pulse program and was oriented by using of Thyreria annulata as the outgroup. Numbers at the nodes indicate the quarter puzzling troop for each internal branch. A scale bar indicates an elementary distance of 0.01 nucleotides per position in the sequence. Vertical distance are for clarity only. The gene bank accession number for the sequences used in the analysis are as follows: Babesia bigemina A, Babesia bovis, Babesia cabali, Babesia diversion, Babesia gibsoni, and Babesia engulfing isolates. Babesia species isolated from Post-tortus and the etiologic agent of infection in 2 cases described here in Europe. And Thyreria annulata is out from the group. Indirect fluorescent antibody technique test, where in this figure the Babesia organisms appear as light green inside the RBCs. Post-mortem examination. Number 1, there are no distinguishing lesions by naked eye either in the aborted fetus or the placenta. Number 2, distended gallbladder can be detected by the sonar before death. Number 3, swollen and friable liver. Number 4, necrotic foci may be noted in the liver in case of Salmonella abortus infection. Number 5, inflamed mesenteric lymph nodes and intestine. Post-mortem examination of the cattle carcasses suffered from babesiosis, characterized by presence of splenomegaly, enlargement spleen, enlarged kidneys, distended gallbladder, enlarged lungs, and enlarged of some lymph nodes as the following: a shape of enlarging subiliac lymph node in equine infected by Babesia equi, enlarged spleen, splenomegaly, and enlargement in kidneys with distended lung of cattle dead with babesiosis, as seen in these figures. Huge splenomegaly and spleen of cattle dead from babesiosis. This figure showing enlarged kidney at the left of the picture and necrosis in glomeruli of this kidney at the right part of the figure. Distended gallbladder as seen by sonar examination before death or slaughtering. This is the shape of distended gallbladder by sonar examination. Differential diagnosis of babesiosis. Cases of babesiosis have to be differentiated from enzootic hematuria, enzootic bovine pyelonephritis, tropical filariasis, post-purulent hemoglobinuria, bacillary hemoglobinuria, leptospirosis, and chronic copper poisoning. Treatment of babesiosis. Treatment of babesiosis is done by imidocar or imizole as 1 mg per kilogram body weight. Dimenazine aceturate is effective as a treatment in sheep, 3.5 mg per kilogram body weight on 2 successive days, or 12 mg per kilogram body weight as a single dose. With supportive treatment include blood transfusion and antihistaminic preparation, anti-shock preparations, and in chronic cases the convalescent patient with hematinic drugs should be provided. As seen in front of you on the pharmacy shelf, you can see the imizole, imidox, and dimenazine aceturate with different trade names. Control of babesiosis. The control of babesiosis in an area depends on the control of tick vector. Number 2, eradication is usually not achievable. Practical because of the high cost to local wildlife, some of which can be hosts to ticks through frequent application of acaricides. Number 3, chemotherapy to kill Babesia in the cattle host and to a lesser degree by immunization of these cattle. Vaccination with live and dead whole parasite, crude parasite extracts, and isolated parasite antigens has been used with varying degree of success. Number 1, live vaccine have proven very efficient and reasonably safe, particularly when vaccination is restricted to cattle of less than 1 year of age, when they are still resistant to the disease. The vaccines are provided either chilled or cryopreserved. They have usually provided greater than 95% protection for the lives of the cattle vaccinated. Number 2, vaccination with subunit vaccines offer an attractive alternative to virulent or attenuated Babesia species. Such vaccines are based on recombinant antigen derived from cloned complementary DNA from protozoan parasites. Several protective antigens associated with merozoites or merozoite-infected erythrocytes of Babesia bigemina and Babesia bovis were identified as a possible molecule. Respiratory associated proteins might become targets of generic recombinant vaccines. Number 3, dead vaccines would overcome many of the inherent difficulties in the production and transportation. However, they have not been sufficiently efficacious and more research is required. Number 5 is the vector control by using of 3 tactic methods as described in the literature. Keep on the track by following us in our technical veterinary learning channel to get the most newest knowledge about any animal infectious diseases throughout the world.